Imagine tiny, glowing sponges, small enough to navigate our bodies. Sponges that could deliver drugs right to a target, or maybe light up cells for medical imaging. It's one of the biggest promises of nanomedicine. But there's a catch. Their glow is incredibly fragile. So today we're going to dive into the science of creating an invisible atomic-level shield to protect them. So here's our roadmap. First, we'll look at the fading problem with these glowing particles. Then, how scientists actually built an atomic shield to fix it. We'll get into the nitty-gritty of finding the perfect recipe and then look at all the evidence they gathered to prove it worked. After that, we'll cover the all-important safety tests and finally, what this all means for the future of nanomedicine. Okay, let's get into it. All right, so let's start with the material at the very heart of this story, porous silicon. It's an absolutely amazing substance, but it comes with one pretty critical flaw. Now, these particles are made by basically etching silicon wafers, and what you get is this incredible sponge-like structure that's just riddled with tiny pores. It's perfect for loading up with drugs. And here's the really cool part. Thanks to a quantum effect, they also photoluminesce. Yep, they actually glow when you shine light on them, which, as you can imagine, is ideal for tracking them inside the body. But here's the deal breaker. The moment these super promising particles hit any kind of water-based environment, like our bloodstream, their surface rapidly oxidizes. You can think of it like a kind of microscopic rust. And this oxidation completely, and I mean completely, kills the glow. They just go dark, making them totally useless for imaging. Now, of course, researchers had tried to solve this before. The old way was to wrap the particles in these thick polymer coats. And yeah, that did stabilize the glow, but it was kind of a clumsy fix. It clogged up the very pores that made the particles so useful for carrying drugs in the first place. So the new idea was much more elegant. What if we could build an ultra-thin, perfectly uniform, inorganic oxide shell? It would be like a suit of armor, protecting the particle's delicate surface without blocking any of its function. So how do you build this kind of ultra-thin armor? Well, to do that, scientists turned to a seriously high-tech manufacturing technique. It's a process called Atomic Layer Deposition, or ALD for short. And honestly, the best way to think about ALD is like you're painting with atoms. Instead of spraying on a thick coat of paint, you are meticulously, carefully building a film one single atomic layer at a time. This gives you an unbelievable level of control, and the result is a perfectly uniform coating that wraps itself into every nook and cranny of a complex surface. So here's the play-by-play. You put the particles into a vacuum chamber. First, you send in a puff of gas. The first ingredient, a titanium precursor. It flows over the particles and a single layer of molecules grabs on and bonds to the surface. Step 2, you purge the chamber with an inert gas, flushing out any extra molecules that didn't stick. Then comes step 3. A pulse of the second ingredient, just water vapor, is sent in. It reacts only with that first layer, which completes one perfect layer of titanium dioxide. And after another purge, the cycle's complete. You just do this over and over again until the atomic shield is the exact thickness you need. But you know, just because the technique is that precise doesn't mean it's easy. The researchers had to figure out the exact right recipe to make this work without accidentally destroying the very thing they were trying to protect. And here's the big irony of the whole process. Water is a totally necessary ingredient in the ALD recipe to form that titanium dioxide shield. But as we just learned, water is also the enemy. It's the very thing that rusts the porous silicon and kills its glow. So you see the challenge. They had to find this perfect balance. Just enough water to build the shield, but not so much exposure time that it puts out the light. And this graph tells the whole story. It so clearly shows the difference between success and failure. You can think of it as a troubleshooting guide. Just look at sample A2. That green line, that was an early attempt with longer water exposure times. And the result? The line is almost totally flat. The particle's glow was just gone. Now look at the blue line, sample B2. By carefully optimizing the process, shortening those water pulse and purge times, they got this beautiful, strong peak. It's a perfect illustration of how a subtle little change in the recipe was the absolute key to success. Okay, so with that optimized recipe in hand, the next step was to gather all the evidence to prove, without a shadow of a doubt, that their atomic shield was not only there, but that it was also doing its job. All right, let's look at the proof. On the left, we've got the chemical fingerprint from a technique called EDX. That really sharp peak labeled Ti for titanium, that's the smoking gun. It confirms that the core component of their shield is right where it should be. Then, on the right, a transmission electron microscope image gives us the visual confirmation. You can literally see that the coating is perfectly uniform. And, this is crucial, the 10 to 20 nanometer pores are still wide open. The armor is on, but the sponge can still do its job. And the numbers tell the exact same story. This data shows the average particle size went from about 200 nanometers to 250. That makes perfect sense. You'd expect them to get a little bigger after adding a coating. But the really critical column here is quantum yield. That's just a measure of how efficiently the particle glows. The uncoated particle was at 0.8%, and the coated B2 sample is right there at 0.5%. This is a huge deal. It proves the atomic armor didn't just stabilize the particle, it actually preserved the delicate quantum effect that makes it glow in the first place. But this right here, this is the ultimate test of durability. The particles were dunked in a solution that mimics the fluid environment of the body. And as you can see, the uncoated particles, well, they faded into darkness within just a few days. But the coated particles, their glow remained incredibly stable for more than 5 months. That is a monumental leap forward, and it proves the armor works exactly as it's supposed to over the long term. Okay, so the particle is stable, it glows, it's looking great, but for any material to even be considered for use in nanomedicine, it has to pass the most important test of all. Is it safe for the human body? So to find out, the researchers introduced the particles to human dendritic cells. You can think of these cells as the key sentinels or security guards of our immune system. And these images confirm that the cells do in fact take up the particles. You can see those dark clusters of nanoparticles right inside the cell, which means we absolutely have to know how the cell is going to react to them. And the initial results were actually very promising. On their own, the particles were found to be non-toxic. They didn't seem to damage the cells or cause them to release these harmful stress molecules. Even at high concentrations, over 90% of the cells stayed alive and healthy. And, by themselves, they didn't trigger a major inflammatory alarm. So it was all looking like a green light. But science is all about the details, right? So they asked, what happens if the body is already fighting an infection? To test this, they added a bacterial toxin into the mix. So, look at the chart. The particles alone, barely any inflammatory response. The toxin alone, okay, a medium response. But when you put the particles and the toxin together, wow, the response was significantly higher. This means the particles act as an immune amplifier. They kind of prime the cells, making them hyper-responsive to other threats. So let's put all these pieces together and really see what this important breakthrough means for the road ahead. This quote from the researchers themselves really sums it all up perfectly. The work proves these coated particles are incredibly promising, but it also uncovers this new layer of complexity. Now, this ability to amp up an immune response isn't necessarily a bad thing. You could even find ways to use it, but it absolutely must be understood and taken into account. So here's the bottom line. Atomic layer deposition proved to be a total game changer for this application. It successfully created a suit of atomic armor that solved this huge, long-standing stability problem for porous silicon. It protected its glow for months in biological fluids, all without clogging up its useful porous structure. And that leaves us with a really fascinating final thought. This research shows us that just making a material non-toxic isn't enough. The next frontier is designing materials that can intelligently interact with our incredibly complex dynamic immune systems. It's not just about being inert anymore. It's about being compatible in a much, much deeper sense. And that is a challenge that's going to define the future of medicine.